Hello everyone, this is Geethanjali. In the previous video, we learned about the Human Genome Project, and in this video, we'll be learning about the DNA fingerprinting. So, DNA fingerprinting was a very useful technique developed by Alec Jeffreys. So, this technique was mainly based on this term called polymorphism. So, what is actually DNA polymorphism? DNA polymorphism is nothing but the presence of repeated DNA sequences. So as we learned in the previous video that most of our DNA genome, the DNA sequences in our DNA genome are repeated. They are repetitive. So in this repetition of DNA sequence is called polymorphism, and as this repetition is inheritable, okay, so this repetitive DNA is also inherited in the future offsprings. That's why in a population, when the number of repeated DNA sequences is similar, we can say that they are genetically related. Now you see, repetitive DNA is divided into two. One is highly repetitive and one is moderately repetitive. So the highly repetitive DNA is basically called the satellite DNA, and the moderately repetitive DNA is divided into further two parts, that is the tandem repeats and interspersed retrotranspones. So in this moderately repetitive DNA, this interspersed retrotranspones means that they are repeated but they are not next to each other. So there's some intermediate DNA sequences that is present between these specific DNAs that repeat themselves over. But tandem repeat means these DNA sequence, they repeat and they are right next to each other like this. Okay, so there is no other DNA sequence between them. This tandem repeats is further divided into multiple copy genes, mini satellites, and microsatellites. So our focus is on these two, mini satellites and microsatellites. So mini is bigger than micro, and micro is extremely small. You can see just dinucleotides means just two nucleotides, right? And mini satellites are more bigger, so their sizes can range from, as you can see here, 0.1 to 20 kilobases. Okay, so the ones coming under this category, mini satellites, are called the VNTRs. So the VNTRs, they stand for variable number of tandem repeats, and this part of the DNA, that is the VNTRs, were used in the DNA fingerprinting technique. So how exactly is this technique done? So first is the isolation of DNA. So how is DNA isolated? It is isolated by the help of restriction endonucleases. So we know that these endonucleases, they cut the DNA. These are molecular scissors, right? So they cut the DNA, and these DNA fragments, they are separated by electrophoresis. So electrophoresis is a technique which we use to separate in DNA fragments that we will learn in biotechnology. And the next step is transferring of the separated DNA fragments into synthetic membranes such as nitrocellulose or nylon. So this process basically refers to blotting. So what exactly is blotting? Blotting is transferring of any sort of biomolecule like DNA, RNA, etc. into some onto some surface or some carrier. So in this case, we have synthetic membranes like nitrocellulose and nylon. Next is hybridization using labeled VNTR probe. So what is labeled VNTR probe? So a probe is a DNA sequence that is complementary to the VNTR, and these are labeled so that we can know that for this specific VNTR, its corresponding probe is this one. Hybridization means basically this VNTR probe will go and bind to its corresponding VNTR. So later, these fragments are detected by autoradiography. So wherever this labeled VNTR, so these labeled VNTR, they are radioactive. There's another reason we call it labeled. So these are radioactive, and that is why we use this method of autoradiography to detect these hybridized DNA fragments. So when we use this technique, the result will be something like this. So let us say there is a crime scene and we need to find out which one is the culprit here, the individual A or the individual B. So we know that when the DNA is same, so the number of VNTRs that will be found will also be the same. So that is what we have to do here. So we take the VNTRs and we perform this technique. Then finally we get something like this. So a particular DNA sequence, that is the particular VNTR, has repeated 2 times here on this chromosome number 16 on one of its chromatid, and you can see in the other one it is repeated for like 5 times. Okay, so if you go to the same corresponding chromosome 16 of the samples, of the DNA samples, you will find that here it is repeated 5 times. So yes, there is a similarity here. But if you see the other chromatid, you will find like 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 instead of 2. So here you can see it is exactly the same. Now which is more similar, this or this? So obviously this is similar because this is the exactly the same, right? So like this, if you compare the other chromosomes too, so you'll find the same number of VNTRs in these chromosomes corresponding to these. So we can come to a conclusion that the culprit there, the one found from the crime scene, matches with individual B, so individual B is the culprit. So like this, we can even find out parents or genetically related persons too, because as we are genetically related, the number of VNTRs present on particular chromosomes will be similar. So I hope you understood the basics of DNA fingerprinting.